So today's topic is complementary genes. For this, the scientists, the names of which were not given in the book, have selected the flower color in the sweet pea, the scientific name of which is Lathyrus odoratus. The scientists observed that all the sweet pea plants are having either purple or white flowers. They have taken two plants which are having white flowers, and these were crossed. And to their astonishment, in the F1 generation, they have got purple flowered plants. That means when two plants, both of which are having same color, that is white color, when they were crossed, they got in F1 generation plants with purple flowers. So these purple flowered plants were crossed. The F1 generation purple flower plants were crossed, and in F2 generation they got purple and white in 9:7 ratio. Thus, the complementary gene ratio is 9:7. They have explained it in this way. Here, this is represented by two pairs of genes. And these two pairs of genes are present on two different loci, and hence they are non-allelic. And here there are two genes, C and P. For your remembrance, think C is for color and P is for pigment. Okay? And there are two genes, one on one locus, C and C. And the other is P and P. Okay? What happened here is, according to the scientists, the parental generation shows white flowered plants with capital C, and small p. And another plant showed small c, small p, and capital P genotype. And when these were crossed, they gave rise to gametes capital C, small p, and small c, capital P. When these gametes fertilized, the resultant F1 individual will have a capital C, small c, capital P, small p genotype. And this genotype results in a purple colored phenotype. Based on this, the scientists say is that here there are two pairs of genes, C, P, and P, both of which are necessary for the formation of the purple color. If any of these two is recessive, that means in first case, capital C, small p, it became white. In the second case, the first locus is recessive, small c, and the second locus is capital P. That means it is dominant. In this case also, it is white. What is happening? When both the loci are said to be dominant, then only the purple flower color appears. Okay. And this was explained by the scientists in this way. How? Let us say purple color occurs due to a pigment called as anthocyanin. Okay? Maybe. And here what happens is, there is a precursor substance. Okay, this precursor substance is converted into some substrate. Okay, it is converted into some substrate. There is a reaction like this, and this substrate is again converted into finally the anthocyanin pigment. So this particular reaction where anthocyanin is produced, there are two reactions. Okay, and both the reactions are said to be controlled by enzymes. So let us represent them as E1 and E2. So E1 enzyme is responsible for the conversion of precursor to substrate, and E2 for the conversion of substrate to anthocyanin. Okay, now we know that all enzymes are proteins. All enzymes are proteins, but not all proteins are enzymes. Here, these two are enzymes, so they are proteins. We also know that proteins are coded by genes. Proteins are coded by genes. According to central dogma of molecular biology, DNA gives rise to mRNA, mRNA gives rise to protein. That means genes code for proteins. So in this example, there are two genes, capital C and capital P. That means two dominant genes. The dominant gene C produces E1 enzyme. Dominant gene P produces E2 enzyme. Okay. That means when this is capital C, E1 is produced. Capital C, E1 is produced. Small c, when it is recessive, enzyme 1 is not produced. Likewise, capital P, enzyme is produced even in heterozygous state, because there is one dominant is enough to produce this enzyme, whereas the recessive genotype does not produce this enzyme. So what actually happened here is, in the first case, this is white flowered. That means capital C, small p, where capital C gave rise to enzyme 1. So this reaction is fulfilled and precursor is converted to substrate. And now for the substrate to be converted to anthocyanin, E2 is required. But here the genotype is small p, so that enzyme 2 is not produced. And what happened? At the substrate stage only, the reaction is stopped and no pigment is produced, and it became a white flowered plant. Okay. Likewise, when we come across with this one, the second one, in this small c and small capital P are present. That means capital P can produce enzyme 2. Small c cannot produce enzyme 1. In that case, in this plant, reaction 2, that is from substrate to anthocyanin, can take place because enzyme 2 is present. But here, the substrate itself is not produced. Why? Because E1 is absent and the precursor itself remains, which results in a white flowered plant. Okay. And thus, here, what we know from this experiment is complementary genes. As we know, DNA double helix, how it is formed? Only based on the rule of complementarity. One has A, that means automatically we can say the other strand has thymine. If this is having cytosine, we can say the other one is having guanine. That means for both of them, both the strands are said to be complementing each other. Likewise, here there are two dominant genes which complement each other to produce one phenotypic trait which cannot be produced when they are all alone. That means capital C, if it is present in the absence of small capital P, that means capital C, small p, it cannot produce. Likewise, the other one, capital P, dominant P cannot produce in the absence of dominant C. It results in white. That means here both the pairs of non-allelic genes in dominant state are required for this character to be produced, and hence such genes are called as complementary genes. Now let us see the definition. Complementary genes are how many? Two pairs of non-allelic, because they are present on two different loci. Two pairs of non-allelic. What kind of genes? Dominant genes that interact to produce only one phenotypic trait. That means purple color. When they are dominant, both the dominant are present, it produces a purple flower. So they interact to produce only one phenotypic trait, but neither of them, when present alone, produces the trait in the absence of the other allele. So only one, if it is present in dominant state, the other is in recessive state, this purple color cannot be produced. So with respect to purple color, these genes are called as complementary genes. And the ratio is 9:7, which you have to remember. Okay. And here, some scientists say that white flowers are there here, white flowers are there here. In the first case, capital C, small p is there. In the second case, small c, capital P is there. What the scientists say is, here, the recessive locus is suppressing the expression of the dominant locus, capital P. Likewise here, small p, which is recessive, is suppressing capital C. So there are two pairs of recessive genes having the same effect. They are duplicating each other with an epistatic influence on the other locus. And hence some scientists, based on this interaction, they call this as duplicate recessive epistasis also. Okay. We have heard about dominant epistasis and recessive epistasis, where in dominant epistasis, the epistatic gene exerts its influence in dominant state. In recessive epistasis, the epistatic gene exerts its influence in recessive state. And here, the same epistatic influence is exerted, but by two different genes which are duplicating their action. Why they are duplicated? Why we are saying duplicating? Because the same white flower character appeared in both the cases. Hence, this is also called as duplicate recessive epistasis. So what you have to remember here is complementary genes. This phenomenon is also called as duplicate recessive epistasis. And the other one you have to remember is 9:7. And if you can also remember this particular definition. And coming to the second one for today, duplicate genes. The ratio of which is 15:1. This you have to remember. And This is also called as duplicate dominant genes. These are also called as duplicate dominant genes. So what actually happens here is they have taken a plant. Its scientific name is Capsella and it is commonly called as shepherd's purse. Okay, its scientific name is Capsella and it is commonly called as shepherd's purse. Shepherd's purse has fruits which are of two types with respect to their shape. One is said to be triangular in shape as shown here. The other is elongated or oval or ovate in shape. So the fruit shape in shepherd's purse, the scientific name of which is Capsella, is of two types, triangular and ovate shaped. We also call it as top shaped. It looks like a top that we play. Okay, top shaped. What the scientists have done is they have taken the plant with triangular fruit shape and crossed it with the plant having ovate fruit shape. And to their astonishment, what they found is they found all the 16 out of 16, all the out of 16, all the 15 are said to be having only triangular fruit shape. Only one is having the top shaped or ovate fruit shape. And this is explained by the scientists in this way. They say that it is again due to the interaction between two pairs of non-allelic dominant genes. Because it is a dihybrid cross, obviously we require two pairs of dominant genes, two pairs of genes. And here also these are two pairs of dominant genes which duplicate their action, So we can represent them as T and V. T and V. So on first locus, there are capital T, and the other locus, there is capital V. And here, the first locus, capital T produces, what produces it? It produces the triangular shaped fruits. And to their astonishment, they also say that as all the 15 are said to be triangular, the second locus also produces the same effect. It also produces the triangular shaped fruits. Thus, all the possible combinations of genes, capital T, capital V, it is triangular. The second one can be capital T, capital V, small v. It is also triangular. Capital T, small t, capital V. This is also triangular. Capital T, small t, capital V, small t. This is also triangular. That means here, when both the dominant genes present, it is triangular. Let's see the other combination also. Capital T, small v. This is also triangular. Small t, capital V. This is also triangular. That means capital T alone can produce the same shape. Capital V alone can produce the same shape. When all these are said to be recessive, small t, small v, double recessive, then only the ovate shape appears. Thus, here in this case, there are two genes, T and V, in dominant state. They produce the same phenotype. In other words, they duplicate each other in dominant state. And hence, such an interaction is called as duplicate gene interaction, the ratio of which is 15:1. And coming to the final topic for today, duplicate dominant genes with cumulative effect. So what is duplicate dominant genes with cumulative effect and its ratio? We'll see now. The ratio of this is 9 is to 6 is to 1. You have to remember this. Duplicate dominant genes with cumulative effect 9 is to 6 is to 1. What is cumulative? Additive. Cumulative means additive. There is 2, 4, 6. So when there is cumulative, what we say is we add all these. 2 plus 4, 6. And when 6 is added, 6 plus 6, 12. And so on. So 2, 4, 6, 12. Likewise, it is added in cumulative manner. And here also, the characteristic feature shows the same pattern. When the dominant condition, homozygous or heterozygous, anything, at either locus, but not both, produces the same phenotype, the F2 ratio becomes 9 is to 6 is to 1. And the example is coat color in pigs. Let's see the coat color. Okay. And here, the coat color is said to be determined by two genes present on two different loci. One is capital S, the other is capital R, capital R. Okay. When the first locus is dominant and the second locus is recessive, as here, the coat color is somewhat like the color of sand. So it is called as sandy color. When the first locus is recessive and the second locus is dominant, what is happening? Still, it is said to be the same phenotype, sandy. So capital S, smaller, smaller, and smallest, capital R. Both are said to be having the same phenotype. So here, when the dominant condition, either homozygous or heterozygous, at either locus, but not both, at either locus. That means capital S on one locus, small r, the other locus, homozygous. Or capital S on one locus, small r on the other locus. So both the loci should not occupy dominant genes. One locus should occupy dominant genes, the other locus should occupy recessive genes. The dominant genotype can be homozygous or heterozygous, but the second locus should be homozygous recessive only. Then only what happens here? They produce the same phenotype. That is sandy phenotype. Okay. So what happened here? Sandy coat color may also result from a non-allelic dominant gene R. It also results from S. Its homozygous recessive is also white. Likewise, gene S can produce sandy, but small s results in white color. When a sandy pig is crossed with a second sandy pig with, here SS is dominant, RR is recessive. In the other case, SS is recessive, RR is dominant. The F1 offsprings were found with, see here, red colored coats. So here these are red colored. Why? The first locus is occupied by dominant and the second locus is also occupied by dominant. That means when both the dominant genes are present, this sandy will not be produced. Okay? And it results in a red color. So in all these combinations, where there are two dominant genes on two loci, all these are red in color. All these 9 are red in color. And coming to the other one, sandy. How sandy appears? For sandy to appear, this should be the combination. Capital S, that means one locus should be dominant, the other locus should be recessive. This results in sandy variety. The first locus can be homozygous or it can be heterozygous. Capital S, smaller, smaller. In that case also it is sandy. Likewise, capital R, smallest, capital R, capital R. This is also sandy. Smallest, capital R, smaller. This is also sandy. Thus, only when either of these loci occupying the different loci are said to be dominant, then only it becomes what we call sandy. So these three and these three. And coming to the last one, where all the loci are said to be recessive. We know that when capital smallest is there, it results in white. When smaller is present, it also results in white. Thus, there are 9 red as seen here, 6 sandy, the blue ones, and 1 white, that is here. And what the scientists say is, to produce a red color, here there is a cumulative effect, additive effect of these two genes. And hence, this type of interaction is called as duplicate dominant genes, which are giving rise to the same phenotype. So duplicate dominant genes, but they have a cumulative effect when they are combined together to form the red phenotype. And hence this is called as duplicate dominant genes with cumulative effect, and the ratio is 9:6:1. So thus we have discussed in this non-allelic interaction of genes, different types of genetic interaction. One is supplementary gene, complementary gene, and epistasis, dominant and recessive epistasis, and duplicate genes and duplicate dominant genes. So let's have the summary. The classical Mendelian dihybrid cross phenotypic ratio is 9:3:3:1. Okay. And for dominant epistasis, the ratio is 12:3:1. And for recessive epistasis, the ratio is 9:3:4. For duplicate genes with cumulative effect, it is 9:6:1. And for duplicate dominant genes, that means shepherd's purse, where triangular seeds are produced 15 in number, it is 15:1. For duplicate recessive genes, that is complementary genes, flower color in Lathyrus odoratus, the genotype, the phenotype is 9:7. And for the another type of interaction, that is dominant epistasis, the same one, here it is again 13:3. And supplementary genes we already have seen. I did not mention here. You can write it down. Recessive epistasis by supplementary genes, the same ratio 9:3:4. I will share this entire what we call, only will share. Summary, you can go through it. Sorry. So you please go through this slide once and try to remember these different types of ratios. And you can observe here. Here, this is one type of genotype where at least both the dominant alleles are present. All four or at least two. And here this is second type of genotype where the first locus is dominant and the second locus is recessive. And this is the third genotype where the first locus is recessive, second locus is dominant. And in the last one, it is a double recessive. When you observe this, in classical ratio, at least one dominant gene should be present when it is present, and the number of phenotypes are 1 and they are 9 in number. Likewise, here this is 3 and this is 3 and this is 1. You can observe it from any dihybrid cross ratio. In dominant epistasis, it is a combination of these two. All this type of genotype as well as this type of genotype combinedly together up to this 9 plus 3, 12 it gives rise to. And the second one is same, 3 and 1. And recessive epistasis or supplementary gene interaction. Here, the AB genotype, that means both dominant genes, genotype is said to be same 9. And these two, that means either the first locus is dominant or the second locus is dominant. Combined together, 3 plus 3, it gives rise to 6 and double recessive is 1. In duplicate genes, what happens? All the 3 genotypes except the double recessive is said to be 15. Thus, we have mixed all these 9 plus 3 plus 3. All these are said to be 15. And coming to the complementary genes, duplicate recessive epistasis. For this genotype, there is 9, whereas whether it is one locus dominant or second locus dominant or both loci recessive, all these are said to be 3 plus 3 plus 1, 7. Likewise, in duplicate dominant epistasis, here, this one is said to be, that means 9 plus 3 together form into 9 plus 3 plus 1 together form into 13, and this one forms the 3. So these are the ratios which you have to remember in this fashion. The slide is already shared and you can try to remember it. Okay. Any doubt with respect to this non-allelic gene interaction, you please ask me. We have discussed about how many? First one, Cum's in false. Second one, dominant epistasis. Third one, recessive epistasis. Fourth one, supplementary genes. Fifth one, complementary genes. Sixth one, duplicate genes. Seventh one, duplicate genes with cumulative effect. So 7 different types of non-allelic interaction we have studied. Any doubts in this, please ask me. Otherwise, we'll move to the next topic. Any doubts? Nobody is there?
There, sir.
Okay. So shall we move to the next topic?
Yes, sir.
Okay.
Yes, sir.
So next, thank you. Just a second.